first slide in your box should be starting with epithelial tissue. I don't know if I can get over here close enough for you guys to see. All right, so remember, epithelial tissue, we're looking for these cells are going to be touching each other. There's no blood vessels. If the cell layer is multi-layered, it is potentially stratified. If it's single layer, it's simple. After we establish that the cells are multi or single, then we want to start looking at the cells themselves. Okay, so you're trying to look for tubes, air holes, or kind of like where something would end and airspace or water or blood or something would flow. So when you're looking at the first slide, it's supposed to be lung tissue. So you're trying to see air sacs, alveoli, and in those alveoli, again, it loses something in translation, but you're trying to look for cells that would line these air sacs. And because we want those cells to be as small as possible, so the distance that oxygen has to move is as small a distance as possible, this should be simple squamous epithelium. Okay, so did y'all find it in yours yet? And then, again, if one group goes with slide 1, the other group can start looking for slide 2, which will be simple cuboidal. So you're looking for slide 1 or slide 2, and they're probably out of order because they're a hot mess in there. Okay, so if you're looking for at slide 1, again, it should give you this kind of open space image. And then if you go to your low power, again, you want to try to find that space where there's openness. And then if you go to your high power, the 40x.
I will contact them, so this is probably going to be different from you, but I can see something.
All right, so again, you're trying to find these like squashed kind of cells lining the opening.
I don't know if you can see something, I just know by imagine that. There's that one.
Okay. Now, if you're not used to using the microscope, start on the 4x. Raise your stage until the little piece of tissue comes into focus, then stop moving the stage, and then use the fine control. Yes, Rebecca.
That was probably not a great question. Yeah,
So
that one's the finest
when
one.
I looked at this, I was thinking this was on the cell.
No, the cells are in there. There's multiple potential cells in there. Yes. Okay, after you find your image under the 4x, you'll go to the 10x. That's your low power, and then you want to readjust just the fine, small adjustments to bring it back into focus. Then you'll go to the 40x, which is your high power, and again, small. If you have a steady hand, you can take pictures with your device through the microscope and get whatever you're seeing. There should be a little pointer in your right eye, so if you want to put that pointer on something and try to take the picture with the pointer there, you should be able to capture that. Okay, so let's talk about, while you're looking at it through the microscope, some of the models that we have simple squamous epithelium. So remember, simple squamous epithelium is lining your blood vessels. That's where blood is always touching these cells. This model is our different blood vessels. So this yellow layer of cells that make the valves in our veins, that touch the blood in our arteries, that then is represented as simple squamous epithelium. So if you look at it one way, they look like a bunch of flat little pancakes, little honeycomb motif. And then if you look at them in cross section, it's a thin little layer. And then it shows you that areolar connective tissue is kind of the gray background, and you don't really see a lot of proteins in that gray because it's a lot of open space and water where blood vessels, actually capillaries to feed these guys, come in. Okay, so this is one model. When we refer to the simple squamous of our blood, we usually refer to it as endothelial cells. So that's one of the ways we refer to simple squamous epithelium in the cardiovascular system. The parts that are lining and touching blood are endothelial cells. Okay. In another model, we have some, as I throw them away. So this is, again, in the kidneys. This is our endothelial cells, and they're showing them creating this capillary bed, and they're showing them as being, again, flattened and kind of long and forming the blood. Then you see another set of simple squamous epithelium. These in our nephrons, in our kidneys, form the podocytes. And you can see they're nice and flat, and then their cell membranes kind of make a little puzzle piece with each other, and that's part of the filtering process. So on this model, it's showing you some more simple squamous endothelial cells, and then it's showing you different simple squamous on top of the endothelial cells known as your podocytes. Okay, and then in the nephron capsule, these cells, and they don't look quite smashed, but these are supposed to also be simple squamous epithelial cells in the circular portion. Okay. Now, the last model, or actually both of these models, all right, this is the stomach model. So what should be the outermost part of the stomach? The visceral peritoneal membrane. So the visceral peritoneal membrane, remember, has mesothelium, which is simple squamous epithelium, and a little bit of connective tissue behind it. So when you look at this model, you can look at it over there, there's a little layer of flat brown cells and then some connective tissue. Those simple brown flattened cells are simple squamous epithelium. We'd also refer to them as mesothelium. And then in theory, if this part is the peritoneal chamber, then the parietal membrane would have simple squamous epithelium and connective tissue. Okay, so that's shown here, and then it's somewhat shown a little bit on this model as well, that here's a little bit of pink, simple squamous epithelium, and then that underlying connective tissue, because this is your visceral peritoneal membrane. Okay, so you've seen pictures in your lab manual. Hopefully we've gotten to a place where you've seen some slide image. And now you've seen it on a few different models. We've seen it in a few different models. Okay, simple squamous epithelium. You're looking for that pancake, that flatness. Okay, now if you haven't seen it yet, let's pull up slide number 2.
You can see that these are a lot more, there's not much open space. These ones are kind of all touching. You can go between the two if you like. It's like a simple.
Oh, get my stage up high enough.
Yeah.
Come on.
Got to the air side.
I'm coming.
Yeah.
Okay. All right, so simple cuboidal, the slides that you tend to have are going to be a kidney and cross section. Okay, and what you're seeing in these tubes, a lot of tubes, right? is we're going to see a lot of tubes from the nephron. Okay, and I'll show you, it's on this model. So imagine we are looking at, again, cross section of a lot of these tubes. Okay, so what you want to see is in the kidney, many of the tubes of the nephron are going to be lined with simple cuboidal cells. So in this part, the nephron is blown up. We know that these are simple squamous because they are around the capsule, but where the tubing of the nephron starts, this is the proximal tube, it is full of simple cuboidal. And you should see there's a difference. They're a little more square. All right, and then this part of the tube is the distal tubule. And again, these should be simple cuboidal cells. So when you're looking at the slides, you're trying to find, all right, I want to try to dig into some of this area that has all of these tubing. So I go from low power, or from scanning to low power, all right? And then I can start to see, okay, there's some lining of the cells. There's a little bit of open space. And the cells typically lining these tubes are supposed to be squarish. Okay, so that would be my simple cuboidal epithelium. So now you've seen a place where it's on the model, on the kidney model, there's at the proximal and the distal tube has simple cuboidal. We're seeing proximal and distal tubes in our slides. So again, another place where we have simple cuboidal. Okay, and then you have your pictures from your book and your lab manual with simple cuboidal. Okay, all right, let's go to the next one. So one of you can pull up simple columnar, the other person could start bringing up the pseudostratified, so we can have some comparison. Okay, so where is our simple columnar mainly? We tend to think of the stomach, the small and the large intestines. So the slide you get is like it's an amphibian tube from the small intestines. Okay, and it shows you simple columnar cells, and it does show you some like little goblet cells that look a little bit whitish and kind of spit out. All right, so those are simple columnar, and if we were to be able to go deeper, they are going to have a lot of microvilli. They're going to make its membrane have lots of hills and valleys because they want to put a lot of proteins on their membrane to be able to pull in a lot of nutrients from the food. Okay, now by model, this is a stomach model. So what should be these purple cells pushing into the lumen of your stomach? Simple columnar cells. Okay, so all of these purple cells here, these are simple columnar cells. And in the stomach, they are going to dip down and make these kind of invaginations, and then they draw them a different color because some of those cells are going to secrete hydrochloric acid, some are going to secrete enzymes. So they kind of specialize, but they're still simple columnar cells forming glands, and then as needed, they will divide and multiply and potentially move up and replace these simple columnar cells up here that are exposed to those enzymes and exposed to that 2 to 3% acidic environment and are in need of constantly being replaced. Okay, so these are simple columnar, right? They don't really show the goblet cells as well on this model. This model is the esophagus. The esophagus is going to have stratified. Then it's the stomach, the small, and the large intestines. So if we look at segment 2, segment 3, and segment 4, the pink cells should be showing you simple columnar epithelium. They kind of look squarish because, again, artistic interpretation, but these should all represent simple columnar epithelium. Okay, so whenever you're thinking of the GI tract, we should be thinking of simple columnar epithelium. All right, those cells on there. Now, did everybody find that? Now, if the other person has slide 5 out, slide 5, well, we'll get back to 4. I gotta find it on here. Come on, where are you? Oh, there it is. Okay. So if you go to slide 5 and you're trying to find your tissue. Now, this tissue that we took a slice of is part of your respiratory lining. Okay? So we took the airway and we made a slide out of it. Okay? Now, the difference here is if you were to be able to get clear images of a simple columnar segment, the nuclei tend to line up. Here, the nuclei can kind of range a little bit because I like to think of these cells as like they kind of weave around each other, and as they do, the nuclei can kind of get pushed up and around, and that gives the impression that they're multi-layered, but they're really single. Okay? And then usually they have some very strong projections of not only their membrane, but cytoplasm and inside the proteins, cytoplasmic proteins that create cilia. And cilia, remember, because of those proteins being in there, can beat. And then they'll have goblet cells, and the goblet cells make a little bit of mucus, and the cilia can move the mucus from the deep parts of your airway back towards the trachea, back towards the larynx, so you can swallow that mucus. And it doesn't get into the deeper portions of your lungs and potentially let bacteria and stuff grow in it and then form pneumonia. Okay? I do not have a model for pseudostratified ciliated columnar cells. So if I'm going to ask you about pseudostratified ciliated columnar, I have to do it by slide or by picture. Okay? Because there's no model. All right? And because most of the time it's with the airway, usually you'll start to see, like if it's the trachea, they'll start to be, later on when we do cartilage, we get a slice of the trachea. So usually a bunch of cartilage will be somewhere around here. And you'll notice that if there's cartilage and it looks like they're arranged in a ring, and then there's an opening and it looks like it's simple columnar, it's really pseudostratified, ciliated columnar. Okay? Okay, so those should be all of our simple tissues that we have in our boxes. So now let's go to the stratified. And if you go back to slide number 4, that's going to be your stratified. Here you go. And so usually we grab skin to do stratified squamous epithelium. And again, we want to try to find a margin where it looks like there's some type of opening. And then what you see in that opening is a pretty dense kind of few layers of cells. And then there's no blood vessels in there. There's no kind of like breaks or tears in them. And they're forming a pretty good barrier. Okay. Underneath is going to be where we see the blood vessels, where we see the features and the different accessories that provide those stratified squamous cells nutrients. Okay, so in our models, the skin model for chapter 5 shows us the stratified squamous layers as this kind of purple layer. And then in here at the thick area, it shows us even the stratum basement. It shows us a little bit of the granulosum and the spongiosum. Then it gives us that white line to represent the lucidium layer. And then the pink outer part would be the corneum layer. Okay, so remember the epidermis has, from chapter 5, they divide it further into 5 distinct layers. The innermost where cell division, that's where your melanocytes and even some of your neurons might try it. Nervous tissue pushes in to get as close as they can to the surface. Okay. All right. You also see in the esophagus, so the first part of this model, we see that it's multi-layered. So there's another one over there and there's another one down there. So on this model, the esophagus would be stratified squamous epithelium. These cells versus the skin, one of the big differences, these are not rich in keratin. Because we wouldn't want this to be dry and repelling water, because that would make it more prone to be cut and break, you know, with things we swallow. So these would not be known as keratinocytes, but these would. Okay, so this is stratified squamous in the esophagus. This is stratified squamous in the skin. This is more keratin rich. This is non-keratinized. Okay, these cells in the skin, we would call them keratinocytes because they make keratin. These we would not. Okay. All right, are we finding this? Okay, the next one should be transitional epithelium, if you have a group that's pulled that up. All right, so for transitional epithelium, this is pretty exclusive to the urinary system. So once on our kidneys, we get to this place where we have the renal pelvis and it becomes the ureters, and then it's going to the bladder. Your urine is made, and your urine is pretty acidic. So we want to keep that urine kind of a little bit more robustly separated from the rest of the body. So from the renal pelvis through the ureters to the bladder, it's going to be lined with transitional epithelium. And the reason why it's transitional epithelium is those tubings and those organs can stretch and open a little bit more to accommodate bigger boluses of fluid. Okay, so this is a ureter, and you can see the ureter is not a hollow open hole. There is some push in, and again, there is some muscle layer that can dilate and stretch. So that transitional epithelium, those cells are going to, when they're not stretched, they are going to look pretty cloudish, big and poofy. Okay? But when they are stretched, they may flatten out a bit. All right, so this is under 40x magnification, trying to show that these cells here are kind of poofy-ish and they push in. There's kind of a nice, you can see a nice division here where the transitional epithelium kind of ends. There's some connective tissue, and then you can kind of see smooth muscle here. Okay. All right, so that should be all of the epithelial slides that we have. Questions? Okay. Now we're going to go into that areolar connective tissue. So we're going to jump to connective tissues. So again, our areolar connective tissues should be anywhere epithelial tissues ends and connective tissue starts, because it is a place where we want to make sure capillaries have the ability to get into. So again, going back here, that gray layer would be areolar connective tissue. Looking again in here, some of this material right behind the cells would be areolar connective tissue. Okay? All right. You would not probably be able to tell me what's collagen fiber versus elastin fibers, but you know the cells in here are fibroblasts, fibrocytes, mesenchymal stem cells. There can be adipocyte. There can be any and all of the white blood cells. The fibroblasts or fibrocytes are there to lay down collagen, elastin, and reticular fiber proteins. Okay? And because there is open space, there's room for capillaries. There's room for small things, small individualized things to make its way through. In that space, if an adipocyte doesn't take up residence, fluid's sitting there, and that fluid is known as your extracellular fluid, and it's also known as your interstitial fluid. Okay? All right? All right. Now, another one of your loose connective tissues is adipocytes. And I like to, when I do an image, I like to try to grab so you can see. Like I like there to be like a little bit of blood vessels so you can kind of see like. Okay, so in this image, like here's a blood vessel right here, and if you look at it, look, there's one squamous cell, there's another simple squamous cell, so you can see some of the endothelial cells here. Out here, there's kind of these open spaces. Those are some of your fat cells, and every now and then again, there'll be some material that collects kind of in the corners of where some of the fat cells are together. Okay, but those fat cells represent your adipose tissue. Right? On our models, our best model of adipose-rich tissue is the bottom of our skin model, which is where our hypodermis or our subcutaneous membrane, is located. Okay? Remember, fat is going to take up residence in almost any and every connective tissue. So these are hearts that my students are going to use on Friday. When you look at them, you see all that white crap in the middle of some of the indentations that the muscle's creating? A lot of that is fat. So this white component in between the muscle, a lot of that is fat. That's not necessarily a good thing. We don't want a lot of fat in some of our connective tissues around like our heart and our lungs, because does fat have a lot of give and go? Not necessarily, okay? But they can show up in high amounts of your connective tissues, and as your obesity, as your percentage of body fat increases, think about there's a lot more fat in all of those connective tissues around all your major organs. And one of the reasons why higher obesity leads to insulin resistance, those fat cells we're learning are very active at secreting a lot of things that screw up other cells and their ability to respond to insulin and respond appropriately to other signals. Okay? All right. So, next one. And again, I like when you see a lot of fat, it looks in the microscope if it ever comes back. Like you can, so look at those kind of cells in the corner and to the edge, and it's showing you the fat cells. Okay? Now, if this person was to lose a lot of fat mass, those cells would just shrink in size, and maybe some of the space between them could fill with more connective tissue and material, but the number of cells would not go down. They would just become smaller cells. Okay? All right, so going to slide 9. Now we're getting into our dense tissues. And look, you can see a little bit of fat. So it's kind of like, here's some fat, and then there's a little bit of a fibrous connective tissue here that's making irregular, probably making a little bit of like holding the spot for the organ. And then you start to get to a lot of laid down collagen fibers oriented in the same way that is creating your ligament or your tendon. Okay, so this slide is supposed to be a ligament or a tendon. And again, there's a little bit of fat. Then there's probably, again, a little bit of like a dense irregular border. Then you start getting into nice lined up fibers. You see a few cells, a little bit of the black. Again, we don't know for sure how much of it is like stains on the glass or something, or stains on the microscope, but there are some cells embedded in here, but there's not as many. It's mostly just protein material. Okay. Now, one of the difficult challenges is if I look at this and I have nothing to help me, clue me in, I would find it hard to be like, is that smooth muscle or is that dense regular connective tissue? So if this image, something like that shows up, read the question. In the question, there should be a clue that would potentially say, here's the arrow, and it would point and say, identify indicated muscle to muscle connecting tissue. Something, or identify indicated bone to bone connecting tissue. So I'm trying to lead you away from picking smooth muscle. If it was smooth muscle, I would probably say identify indicated movement producing tissue. So there will typically be a clue. The clue might be something about its feature, or it could be maybe where it's located. Okay? Because again, that is a hard thing. You just see pink. And pink is very closely like, is that muscle or not? Okay, so I would give you a clue. All right, so that was number 9. Let's go to number 10. All right, number 10 is cartilage.
Our 10 is broken.
Oh, that's okay.
Okay. Fine.
If it's broken, but it's not broken on the part where the tissue is, you might be able to still put it on the stage and look at it. Okay, so remember that cartilage, you're trying to find a place where the matrix looks a little thicker. There's no blood vessels. And your chondrocytes are living in these little chambers called lacunae. So here in this picture, this area fits those definitions. The matrix seems a little thicker. The cells seem to be sitting in these little chambers, and there's no blood vessels in here. Okay? And I don't know that y'all can see that all well. Now, on the outside, remember that's your perichondrium. So there might be some dense irregular connective tissue here to provide us, again, that capsule, that material that lets the cartilage kind of, again, hold its spot and even, then slowly merge into the hypodermis or whatever other areas it needs to merge into. Okay? Typically, in the cartilage, no white blood cells, just the chondrocytes. And again, because the blood flow only comes into the perichondrium, if you get an injury in here, it's very slow to heal because everything has to diffuse, and these cells have to regrow and fit, and they're not fast-moving cells, and they don't do things at a very fast pace. So that's why sometimes with injuries, if you break a bone, you could recover in 6 weeks. If you tear cartilage, you could take 6 months to a year. Okay. We do not have any models of cartilage. I really don't have good models of tendons or ligaments. I mean, I guess I have these here. Hopefully you would know that these are tendons or ligaments, right? If I've showed this model showed up, okay? Pointing to things connecting bones, okay? So for cartilage, you're going to have to look at it as a picture from the lab manual, from the book, or the slides. Okay, so this is hyaline cartilage. How do I tell my different cartilages apart? I don't really see any large amount of protein. So remember, hyaline cartilage, there could be some protein in there, but there's not a large amount. As the chondrocytes make a lot of elastin protein, then I would get elastin cartilage, and you would start to see more fibers. So if you go to the next one, the elastin cartilage, okay? And again, you're trying like, oh, no cartilage yet, but there it is. Look at that beautiful fat. Look at that beautiful picture, right? You see fat, you see cartilage, and then you see a bunch of dense, irregular connective tissue. Okay? If we go and blow it up, okay, the part of the, so there's some fat up here. Then you kind of see dense irregular connective tissue because this is the perichondrium. Then you start to see the cartilage, and the cartilage, can't see it great here, but it's got a lot of fibers. So the matrix is a little jelly with some protein, and then you start to see again the perichondrium. Okay, chondrocytes sitting in a chamber, no blood vessels. But there's more protein, and so I'm telling you it's elastin protein. Now, can you tell the difference between collagen and elastin fibers? No, because I can't. So if you saw this image, hopefully you at least know it's cartilage. If I asked you to tell me what specific type of cartilage, I'm going to need to give you a clue because this is elastin cartilage. So I have to give you a clue to make you not think it's collagen fibrous cartilage and it's not hyaline cartilage. So one of the clues would be this cartilage has a lot of elastic ability. This cartilage is found in the epiglottis. Okay, this cartilage has to maintain a lot of bendability throughout its lifespan. So it has to have a clue to make you think those stringy proteins in the matrix are elastin, and this is elastin cartilage. Okay, so last cartilage we should have would be something like your vertebral discs. Okay, those discs between your vertebra. There it is. Okay, so going up to low power so we can get a big cross section of this image. All right, so to be honest, it looks like there's a lot going on here, right? And there's nothing that really shouts out cartilage. Okay? So when you're looking at this, again, maybe I would try to find an area, maybe like here, where as you go into the higher power microscope, you would hopefully clearly see those lacunae, and that matrix is, so that's like shredded a little bit. So it's still kind of, my slide looks really damaged. But something hopefully that would show you lacunae and chondrocytes. So my slide here, to me, doesn't do a good job of showing cartilage because it's all shredded up. Okay. So we've looked at 3 different types of cartilage. So usually the presence of the protein is going to be one of the clues that it's not hyaline. If they're looked to be in the matrix, those proteins, then read the question for the clue. If it says this tissue is found in the meniscus or in your vertebral discs, you would then be looking at it going, this has got to be fibrous or cartilage-rich cartilage. Cartilage. Okay. All right. Next, keeping in our connective tissues should be bone. Bone is pretty easy to tell what it is. Okay, so with bone, you're going to have a lot of solid material. If it's compact bone like this, you want to see a lot of these like tree trunks pointing up at you. Okay, and the tree trunks with the circles, the concentric rings, are telling you it's osteons, and the very center should be some blood vessels running through. All the little dots kind of there, those are your osteocytes. And the like white material they're embedded in are your lamellae. And remember, you can kind of see some of them have these protrusions. Those ditches in the lamellae are known as canaliculi. And they are dug by the cell to try to reach closer to the blood supply. Okay? Now, for a model, we have this model. Okay, so again, you're seeing some osteons. The white material, that's the solid matrix, is the lamellae. The cells embedded, and they look like these little ants almost. Those are the osteocytes. The little legs, if you think of them as ants or insects, they are digging in the lamellae, and that ditch or that little tunnel is canaliculi. And those cellular projections are trying to get the cell as close as possible to the blood supply. These cells are in a lacunae because they need to be in water. And so they make a little chamber so water can kind of fill. Okay, now, compact bone running through the osteons are the long ways vessels. Running between the osteons are going to be perforating canals to let the vessel kind of, push through the bone. Like cartilage, the outer part of the bone is technically connective tissue. This is your peri for around your bone. So this here is dense, irregular connective tissue, and then there's a cell layer. And we'll learn that cell layer is a mix of osteocytes that can become more bone or bone making or bone taking away. And then there's fibroblasts, and there can be white blood cells and adipocytes and all that stuff in here. Okay, so remember this collagen in this dense irregular periosteum here, that is what the ligaments and the tendons like bind into, so that way they can join to the bone. Okay. So there's the bone model, there's the bone picture. Okay. Last of our specialized connective tissues is our blood. All right, so again, blood. The background material is 92% water. The cells are known as formed elements. Most of those cells, you count 100, 99.9% of the time, you're going to count 100 red blood cells. In the corner here, there's a white blood cell. See, it has a purple nucleus and some material. And then over here, there's another white blood cell again with a purple nucleus. That's how you tell the white blood cells from the red blood cells apart. No collagen, no elastin. So the matrix in blood is the only place we don't want collagen, we don't want elastin. That's part of the reason why no fibrocytes, no fibroblasts. And then there should be no adipocytes in there either. By model, here it is. Has a lot more white blood cells than normal. The yellow background is your plasma, your extracellular matrix. This is mostly water. There are proteins in here. There are antibodies. There's some clotting factors. There's albumin, transport molecules, and some of your hormones, but you don't see them. They're not big strings like collagen and elastin. So no collagen and elastin. Most of the cells are red blood cells. There are some intermixing white blood cells, immune cells, and then the little blue things represent the platelets. And again, we don't see the platelets with our naked eye. Okay? No fibroblasts, no fibrocytes, no adipocytes, no collagen, no elastin. And this is always, blood is always touching simple squamous epithelium, endothelial layers. All right, hang with me, we're almost done. Okay, next slide, we're now moving into our muscles. All right, so first muscle, skeletal muscle. You have to get the magnification right. You have to get the focus right. You're looking for a long, skinny cell that has striations. The long, skinny cell can have multiple nuclei. The long and skinny cell is technically not touching any other cell. Okay, so your skeletal muscles are technically not touching each other. And they're nice and long and they have multiple nuclei. And you can't see it very clearly because it's lost in translation. But hopefully when you look at it on the microscope, you see the striations created by the sarcomeres. Okay, remember this is under voluntary control. This is usually going to stain red because it's large in blood flow, and inside those cells are a lot of proteins that collect oxygen, and they have iron cores called myoglobin. All right, moving to cardiac tissue. Okay, so cardiac tissue, we should still, under our high power magnification, we should still see the striations. Okay, you have to get them in the right kind of orientation. You want to see them in longitudinalness. Okay, so we should see striations. And then we should see, this one slide, I haven't been able to really find it yet. We should see some intercalated discs. So the pictures in your book show them really well, where one, because remember these cells are single nuclei for the most part, and where one cell ends and another cell begins, you should see some thicker bands. And I can't seem to find them on here. Okay? So those thicker bands, so they're going to stain red because they have myoglobin. They're going to stain red because there's a lot of blood flow in there as well. They're going to have a lot more nuclei, and they are going to supposedly have, where two cells connect, they are going to form a darker band that represents the membrane and all those proteins that are coming together. So when you shine a light, they're absorbing more light energy. Okay? Those are intercalated discs. Then if you look at the cell, you should still see the striations the sarcomeres make. Only found in the heart, are going to be under involuntary control. They have their own pacemaking that tells them to contract. Okay. Next is your smooth muscle. Now you want to probably get the smooth muscle for the intestine slide rather than individualized teased smooth muscles. Okay. Now, remember we have smooth muscle represented in our stomach. You should have smooth muscle in the esophagus, in the stomach, in the small and the large intestines. Typically, smooth muscle is going to run in the tubes and the pipes of our body. It's also even here, represented by that red, in our blood vessels. It tends to run in circles. One layer is a circle, and the other layer runs long. So in our slide here, you can kind of see some of these cells are shooting out at us, and then some of these are running long. Okay, so when you're looking for smooth muscle, you want to find usually tubes. I don't know what happened. All right, so you want to look for, again, usually there's going to be 2 layers of them. The circular layers help you change the diameter. The longitudinal layers sometimes help you kind of mix this way. Okay, so a lot of the gut, again, my esophagus, a little bit of shrinkage helps, again, make smaller amount of distance. Things have to be swallowed. Tubing can help, like toothpaste, push something further down. All right, so smooth muscle. Now, again, it shows red, it shows pink. It's individualized cells. These cells can touch each other. There can be push in by blood vessels. It can be really challenging to try to be like, is that dense irregular, dense regular? Is that smooth muscle? So you want to look for clues. These are contracting cells. These cells they're under involuntary control. They can be told to contract by hormones, local cell factors, the nervous system, and automatically, just intrinsically. Okay, and we tend to find them, again, running in the 2 directions together. Right? No striations. Okay, and then I have another skin, which I feel like we did that one already. Oh, uh-oh. Did it break?
No.
Okay, because I'm going to pick it up with glass. All right, so again, I don't know, I can't see this one all that great. Okay, mine's terrible slide. All right, so if you want to pull out the slide again, all right, wait, I don't have a, maybe it's backwards. We should have a neuron. All right, spinal cord, there it is. All right, so our last tissue after our muscle tissue is our nervous tissue. Okay. And so far, we've been focusing on the neuron. Looks a certain way. It's a big kind of one-eyed monster, and then it has one long projection. So in your spinal cords, all right, in these kind of like butterfly areas in the anterior kind of wing side, you should be seeing a bunch of neurons. Okay, they're cell bodies. All right. And then you see kind of a lot of material around them. That's the neuroglial cells and just the extracellular matrix. Now, my model of my neuron is back here.
Oh, wow.
Okay. So if you were to amplify out, you would want to look at like some of these big things. There's the cell body, and the cell body is where the nucleus and the nucleolus is located. There's a lot of material around it, and then it has a lot of projections of its membrane to allow a lot of neurons to be able to synapse on it. So all these little rubber things are synapting axons on this neuron. Normally you'll start to see one protrusion and it'll quickly like disappear because it gets super, super, super thin. But in theory, that projection, which this would connect here, is your axon. Okay, so this is your cell body. All of these projections that the axons are terminating on are your dendritic areas. The one protruding axon of this is here, and then it shows it here, and it shows it with some supporting cells around it. So these are neuroglial cells, and then it shows some other axons terminating on this guy. Okay, so this is one of our neuron models. Our other neuron model is from the eyeball, and these neurons here, right? So these are our rods and cone cells, and then you can kind of see there's there's some neurons running here, and so they look a little different because they're a different type of neuron, but they are connecting from the rods and cones. They will connect, and then their axons will eventually run as part of your optic nerve. Over here, it shows like a little bit of a diagram. So it shows your rods and cone cells in blue, and then it shows some different neurons that are getting the information from these cells, and then eventually their axons are going to run and be part of your optic nerve. Okay, so not every neuron looks like this, like the book is letting you lead. Some of the neurons are going to eventually have some different shapes, but this is what we've learning so far. So dendrites, axon, cell body. Okay, last thing is the skin. Finally, I can sit down. Okay, so just like the picture from chapter 5, this picture, this model go really well together. Right? So you have epidermis, then you have dermis, and again, where the dermis ends and the hypodermis begins, it's an imaginary line. The big thing that we know is going to be in the hypodermis is a lot of adipocytes, right? And we tend to think that the hair, the bulbs, the roots tend to be more in the dermis. Okay, so you kind of have an imaginary line, right? For that epidermis, again, there is 5 layers, stratum basal, then you know it's the stratum granulosum, stratum spongiosum, then you can see the white line here for the stratum lucidium and the stratum corneum. We know that the dermis and the epidermis of a lot of places in our skin form those hills and valleys to press into each other. So that is known as the dermal papillae, and you can see it here as like the skin's been pulled off. Okay? Stratified squamous epithelium, keratinized, so these cells are keratin rich. They are known as keratinocytes. As they move away, they are going to slowly die. So the dermal papillae helps keep some of them alive a little longer. All right? And remember, in that stratum basal layer, that's where mitosis, a lot of cell division is happening. That's where your melanocytes, that's where some of your nervous tactile corpuscles, and they show them over here, those specialized neurons that have features that are going to be sensitive to different stimuli close to the surface of the skin, they are going to embed. Okay. The other thing about the dermal papillae is that's where your blood vessels tend to get and capillaries. Okay. All right. Moving into the dermis. The dermis, remember, is a mix of a little areolar connective tissue here, dense irregular connective tissue, collagen rich, and reticular fibers to make some big spaces for some of the big features we have. Some of those big features are with the hair pointing out. These are keratinocytes that have died. As you go in, there's a follicle, and then there's the root of the follicle. The hair, the cells that are living and dividing and multiplying are in the root. Okay? And tied to that root, there's an opening where a nerve and blood supply comes in. And so that is how those stem cells in there continue to stay alive and get their nutrients. And that is how all of your hair is eventually tied to this nerve. And so if you do laser hair burning removal, that's why it's painful, is because you're burning that nerve ending so it no longer is attached to anything. because you're killing everything it's attached to. Okay. Many of our hair follicles, as you go up, they will have these accessory features of stratified cuboidal epithelium that make little kind of leaves. Those are your sebaceous glands. They are going to create sebum. And the sebum is going to get pushed into a duct that opens with the hair follicle channel, the hair follicle, the hair material that's being pushed to the surface. And then it'll come out of the same hole as the hair to lubricate the epidermis. Okay. All right. The other thing attached to our hair follicle usually is a little bit of muscle. This is actually smooth muscle. It's erector pili muscle. It will look a little striated, but it is smooth muscle because even though it's striated, it's under involuntary control. And it is what contracts when the temperature and that reflex of let your hair stand up to try to trap air to warm it. Again, if you had enough hair, you would be able to keep yourself warm without clothing. Going back to our chimpanzees and our gorilla ancestry, they still use that to help warm their bodies. We don't anymore. But we have some common ancestor, and this is still remains. All right, so that erector pili muscle. All right, the other thing you see in here are these eccrine or merocrine. So this is the gland, and then kind of like make it 40 times bigger. And it's showing you if I cut away, it's one long tube, usually simple cuboidal cells, all right? And they all secrete in a few solutes and water follows, and then that tube connects to the surface of the skin for sensible perspiration. So that's the perspiration you do for body temperature regulation. All of these cells that are losing their cell water as they die, that water is the insensible perspiration you lose. And you can't control that. You will lose cell water from your body no matter what. This is the one we try to control by not being in extremely high temperatures for extended periods of time, or drinking lots of water to replenish it as it's happening. Okay, neurons up here, tactile corpuscles. Down here, this one would be a laminated corpuscle. Eccrine, merocrine, pseudiferous gland, sebaceous gland, erector pili muscle, hair follicle, hair root, hair shaft. And I think that's everything. So, questions? Okay, so test on Saturday. I would say that other than the chapter 1 material, that is a lot of the test because we've been through every single tissue and the skin model. So add in body regions, landmarks, anatomical terminology, directional terminology, planes, cutting things up, which plane am I cutting in? You know, that's what's not, we didn't cover today. All right, I'm going to stop the recording.